that he's helping us today, and he'll give you a mic that you can speak into, and then I can hear you and answer your questions. So I'll be talking today about deep learning for brain encoding. So far in today's session, we had the introduction to the brain encoding and decoding. We talked about stimulus representations that are needed to do this encoding and decoding. And we also looked at deep learning for brain decoding. So today, right now, in this afternoon session, the first one we'll be talking about the encoding part. So that will be split into two sessions. The first one will be about the classic findings and some of the more common approaches to encoding for learning more about how the brain functions. And then in the second part, I'll be telling you more about some of the more recent findings that actually utilize deep learning. So first off, what do we really mean by encoding and when is it useful? Well, in brain sciences, we're really interested in understanding the mechanistic, we want a mechanistic understanding of how information is processed in the brain. And that really leads to four big questions. So the first one is what information is actually being processed by the brain? When is it being processed? Where is it being processed? So what locations in the brain are actually involved in processing this specific information? And also, how is all of this information put together both in time and in space to give us this really rich understanding of the world? And so at least my inspiration in looking into this, so I'm coming from a computer science background, is that we already have this system that has true intelligence, the human brain. And by being able to model it and study it, we are getting closer to understanding at least one true system that can understand the world and adapt to different kinds of environments. And we can try to emulate that in machines as well to make progress towards a more realistic artificial intelligence. So now we have these four big questions that can actually get us closer to understanding the information processing in the human brain. But actually, how can we move forward on those questions? Well, encoding models, which is what I'll be talking to you today about, are one way to actually learn more about these four questions. So an encoding model predicts brain activity of a person as a function of some representation of the stimulus that corresponds to the specific brain stimulus that somebody is actually viewing. So for example, if you are looking at a picture or you're even watching a movie, and we're scanning your brain response as you're watching this movie. So you're receiving both visual information from the video aspect and also auditory information from all the dialogue in the movie, as well as the different sounds that happen that don't have to do with speech. Then your brain is lighting up in some way that we can capture using, let's say, fMRI, which you have. If you were joining for the previous sessions, you have a little bit of background on this, but essentially it's a way to image the brain response and get a time series of the brain activity as somebody is viewing some kind of stimulus. And so the encoding model can help us understand how, what different properties of the stimulus of the movie you're watching is helping you elicit this brain response. So for example, if you were interested in understanding how the part of speech of particular words is leading to this particular brain response, we can have a stimulus representation that actually encodes this part of speech. So for example, for orbit, this one word here in the stimulus, then the part of speech would be noun. So we can build a stimulus representation that's a one-hot vector encoding of this of all the different parts of speech, and the one will represent the noun, and there will be zeros for every other part of speech for orbit. And so the stimulus representation is actually a hypothesis for what kind of features of the stimulus actually affect the brain response. So these are some latent brain relevant stimulus properties. So we don't actually know what these stimulus properties are that are affecting the brain, but we have some hypotheses for them, and we'll computationally represent them with the stimulus representation. Right, so these stimulus properties are latent, and the actual stimulus representation that we built is some subset of these stimulus properties, if it's a good stimulus representation. So of course, we can also build other stimulus representations that don't actually overlap in variance with the stimulus properties that affect the brain response, but those are not effective stimulus representations. So today we'll be making an assumption that we are building some stimulus representations that are in fact meaningful and represent something about the stimulus that is brain relevant. And of course, we have ways of evaluating this assumption as well. And I'll be talking to you about that. And one way is actually this building this encoding model. So I'm talking a lot about these encoding models. What actually are they? So I'll give you a more specific dive into encoding models in the following slides. But I do want to tell you here about this very high level understanding of an encoding model. So an encoding model learns a function, f, which takes as input the stimulus representation. So for example, this one hot vector encoding for part of speech, and predicts as output the brain activity in a particular region or time point in the brain response. So we have this, for example, this green volume pixel over here. So we learned this function directly from the data, and then we evaluated on some data that we have held out during training. And this is usually done by looking at how similar were the predictions that we made for this particular point in time or location in the brain with the true data that we have not used for training. And that can tell us how well did we actually do at predicting this brain response. So this function f is actually the encoding model. So the weights of this function, and again, I'll tell you a little bit more how we parameterize it and how we learn it in the presentation later on. But just at a high level, this function f is representing the encoding model. So this encoding model can in fact reveal which brain areas are affected by specific stimulus properties that we have represented with the stimulus representation. So using these encoding models, there have been some classic findings about the brain function. So these classic findings do not use representations from deep learning models. In contrast, they use very interpretable representations. So I wanted to just contrast, before I dive into the more recent findings that make use of deep learning, I wanted to give you a specific example of what this kind of findings can be if one uses interpretable features to represent the stimuli. So we'll split this into 3 parts. The first part is about this classic findings in the language domain, which is from a seminal work from Tom Mitchell and colleagues, which was published in Science 15 years ago at this point. And then in vision, there was a quite similar in terms of methods work, which was also very seminal, which was published by Kendrick Kay and colleagues in the other very prominent journal Nature. And then a few years later, there was another work published by Santoro and colleagues in PLOS Computational Biology, which used similar methods for the auditory domain. So we'll touch on one of these studies in each of those 3 key domains that actually use interpretable representations of stimuli and learn something about the brain or confirm the hypothesis about the brain using these encoding models. So let's focus on the language first. So in this work, the authors here used specific stimuli that corresponded to people seeing concrete nouns as well as their line drawings. So here I have an example for you, which is the concrete noun bear along with the actual word bear. So bear word and then the drawing corresponds to bear. So people actually saw those drawings and words for 60 different concrete nouns. And their brain activity was recorded with fMRI. So here, the stimulus representation for this, these kinds of data that the authors chose was a corpus co-occurrence representations, which corresponded to 25 sensory motor verbs, such as see, hear, taste, and smell. And the reason for choosing these 25 sensory motor verbs was a hypothesis from, or a theory, I should say, from cognitive science and neuroscience, which thinks that the way that we represent semantics in the brain is embodied, or in other words, the different attributes, semantic attributes that we use to represent meaning are represented in the parts of the brain that also help us actually process the sensory motor experience that's related to this attribute. So for example, for the concept banana, the visual aspect of banana, the semantic visual aspect of banana would be processed in the visual area of the brain. The way that the banana smells and tastes would be stored in areas that have to do with actually experiencing what the banana tastes or smells like. So here the point is that even without smelling banana, if I just said banana to you, this concept would elicit semantic properties in these different parts of the brain that have to do with actually experiencing, seeing and tasting a banana. So there's empirical evidence for this hypothesis, for this distributed organization of attributes that are related to all these different senses in the brain. So audition and perception of color and shape, motion, and also action and taste. So the authors here chose 25 sensory motor verbs and looked at the corpus co-occurrence statistics of these 25 verbs in large amounts of text data to actually build the representation of the concrete nouns. So now essentially in order to represent bear, the vector that is used to represent bear have the co-occurrence of bear with these different verbs. So see bear, hear bear, taste bear, and so on. So now the authors record brain activity of people seeing these concrete nouns in line drawings, and they're able to build an encoding model that as input this 25 element vector that corresponds to co-occurrence statistics with the 25 sensory motor verbs, and predict as output different XYZ coordinates in the brain that corresponded to people actually seeing these concrete nouns. And what they showed is that for the first time, is that one could accurately predict these fMRI recordings for a novel word. So the coding model allowed you to actually be able to predict the brain activity for, let's say, telephone, even if telephone was never once seen during training. So this is zero-shot prediction of the brain activity while somebody is seeing telephone. And what was even more interesting was that the authors looked at the weightings for the encoding model. So what part of this 25 sensory motor feature space actually contributed to the prediction of different parts of the brain. And they saw a correspondence between the semantic property for let's say, a specific sensory motor verb like push with the function of the cortical region of the place in the brain that they're actually predicting. So they saw that the weighting on the verb push was actually corresponded to where in the brain push was important for, which was the sensory motor cortex, which actually relates to pushing. So this goes really well with this embodied cognition hypothesis that states that we actually store the meaning of these different concrete nouns in a way that matches with how we experience them. So this was a very nice way to use encoding models to verify a hypothesis or provide even more evidence for hypothesis in cognitive science. So this is about this classical finding in language using encoding models. And now I want to tell you a little bit about a classical finding in vision. In here, this is a different set of authors, and they worked with a set of natural images. So this was people who perceived about 2,000 natural images, and the brain recording was taken using fMRI, and the participants were viewing these images. And here, the stimulus representation that was used was a mixture of Gabor wavelets. So here they have a couple of different stages in this work, and in the first stage they actually estimate the encoding model that takes as input this mixture.
Sorry, Mariya, just a second. It looks like we have lost the room. Give me a few seconds. People in the room, can they hear me? I think we've lost the room. Sorry. Okay. Trying to just check with people.
Thanks a lot for noting that. I saw the camera going off, but I didn't realize everything was off.
No, yeah. It just went off a few seconds back. So, yeah, let me see if I can. Yeah, I'm just okay. Let me in fact call up Asif, because they may not even realize it, because they will clearly not be standing next.
I hope it was not because of
awesome. Yeah, Abhiram, somehow
actually the internet here is very weak. Actually, it got disconnected. So right now I'm using a hotspot from my new Chinese SIM. I think it should be fine now. It's far better.
Abhiram, please turn off the video. Turn off your video to save on the bandwidth. And you could, and potentially, Mariya, if you don't mind, maybe you can turn off your video as well.
No, I guess it should be fine now. Actually, it's running on a 5G network, so it should be fine now.
And your mobile SIM has no issues with that?
Oh, I'm turning off my video. Actually, this stream here, you can switch on your video from your end.
Okay, Mariya, please go ahead as you see fit. Yeah.
Okay.
Good. Yep.
Okay, sounds good. I will turn on my video, but then if we encounter another problem with the internet, I can turn it off again.
Awesome.
Okay, sure. Okay. Perfect.
Thanks a lot for making this work. Yeah, we really appreciate it. So can you, am I audible now, and are the slides visible?
All good for me. Yep. I think all good at the room as well.
Hopefully. Okay, great. Great. So I think we cut off at the end of the language part. So here, what I was saying was that now we'll discuss these classical findings in the vision domain, in which people saw a set of natural images. So this was about 2,000 natural images, and their brain activity was recorded using fMRI while they were viewing these natural images. So here the authors chose a stimulus representation that was a mixture of Gabor wavelets. And this is because of understanding that the primary visual cortex in the brain processes edges of different objects and so on. And so the Gabor wavelets were a very good stimulus representation to choose to actually test this hypothesis. So here, the first part of this work was to actually estimate the receptive field for each different voxel in the brain using these Gabor wavelets. So here they trained an encoding model that took as input this mixture of Gabor wavelets and predicted as output different voxels in the brain in V1, so the primary visual cortex, to V3. So still visual cortex, but a little bit higher level than this very beginner early visual cortex, which is V1. And so they used these kinds of encoding models to estimate the actual quantitative receptive fields of different voxels in the brain in these different areas, V1, V2, and V3. And what they showed was that you can actually use these encoding models to estimate these receptive fields for these different visual areas. And they, on top of this, they used the encoding model to actually identify which of a set of candidate natural images somebody was viewing. So this was a big breakthrough at the time. So this is the first time somebody was able to actually essentially identify what stimulus somebody was actually viewing. But of course, this was out of a set of some candidate images. So it was not open domain evaluation here, but still it was a very big step forward for this area. So this was about the vision part, and now the last domain that I want to discuss is the audio domain. And again, this was a few years later in 2014. And here the authors looked at a set of natural sounds, so speech, music, nature, and some sounds from different tools like a drill. And here the participants were viewing these different sounds as the authors scanned their brain using fMRI. And so the participants were listening to these different sounds. And here the hypothesis that the authors were trying to investigate was what kind of spatial and temporal granularity are different brain regions really sensitive to. And so they used a stimulus representation that actually varied along space and time, the granularity along this space and time. So they use spectrotemporal filters that are selective for modulations along this space and time continuum. So here I just have a diagram of a few different conditions that they use to train the encoding models with these different spectrotemporal features. And they use these different filters to build an encoding model that predicts specific part of the brain, which is early auditory cortex. And here they saw different things in terms of how different parts of the auditory cortex is being predicted. So what they saw as a high-level takeaway is that there's a posterior dorsal, posterior anterior divide in which the more posterior regions, or this means the more back regions of this auditory cortex, like towards the back, they are more selective for coarse spectral information, but also high temporal precision. Whereas the more the regions in the auditory cortex that are closer to the front of the brain, the more anterior regions, correspond to more fine-grained spectral and low temporal precision spectrotemporal filters. So this was a very interesting finding, and was one of the first times that encoding models were used to verify some hypotheses and also present new findings in the auditory domain. So these were all, these three different examples were examples of how using coding models with very interpretable features to understand something about the brain. Well, what we're also very interested in now in more recent years is to understand the processing of very naturalistic stimuli. So for example, when somebody is reading a book or is watching a movie. So as you saw in the previous examples, people were observing very individual stimuli. So for example, a concrete noun or one particular sound at a time, like a sound from a car or a sound from a drill, or an individual image that was not contextualized in the broader experience. And so neuroscience and cognitive science is moving more towards using naturalistic stimuli like these reading of a book or watching a movie to really understand how we are processing these everyday stimuli and to help us gain a more environmental understanding of how our brain is functioning. And so using these naturalistic stimuli, now there's a lot more stimulus properties that are affecting the brain activity, because these stimuli are not controlled for any specific part, and they're also quite, there's a lot of things happening in these stimuli. So for example, in the movie, there's the visual stimulus and the auditory stimulus that are affecting the brain activity together. So now simple stimulus representations are not actually able to explain as much variance in the brain as they were able to when we had more simpler stimuli that we were presenting to participants. So now what do? How can we actually build stimulus representations that are more meaningful for these naturalistic conditions? Well, meanwhile, we have had, of course, a huge development in deep learning in the last 10 years, which really gives us AI models that can do many different tasks in very naturalistic settings. And of course, they're not perfect and are sometimes actually quite flawed. But there is the question of whether we can use representations, internal representation of the world from these models as rich stimulus representations that perhaps are capturing some of these properties that are also relevant for processing in the brain. And just as a spoiler alert for the work that I'll be telling you about in a few slides down in this presentation, these representations, internal representations from these deep learning models, like for example, language models, can explain substantial variance in the brain response as somebody is reading a book or even watching a movie. So we do see this quite significant correspondence between the representations in the models and the representation in the brain. So actually learning, using features from these deep learning models can enable data-driven encoding models for these naturalistic stimuli. So how can we actually build this data-driven encoding model to evaluate the relationships between the brain and deep learning models? Well, the way that this usually works is we have some, let's say, naturalistic stimulus that can be multimodal. So it can be somebody watching a movie and receiving a visual and auditory input. And then we present the same stimulus to a deep learning system that can also process similar sensory modalities. So for example, if we have a multimodal stimulus, we would want to use a multimodal model. For example, a video transformer that can take both the video frames as well as the auditory input as input and create joint representations of the two. And we present the same stimulus to a person as we scan their brain activity, as we record their brain activity with different brain imaging devices, for example, fMRI. And then we obtain the representations of the stimulus from the model as well as the brain. So from the model, we can extract internal representations from different layers in the network. And so this is essentially a vector with some real numbers that, for example, for some of the early variants, such as BERT, they were something like 768 dimensional. And then we can also extract representations from the brain, from specific regions in the brain, where we have a hypothesis about which of these regions are involved in processing of this naturalistic stimulus. So if the stimulus was language related, then we would focus on some regions in the brain that we know process language from previous work. So now essentially we have two representations, two high-dimensional vector representations of the same stimulus, one coming from the machine and one coming from the brain. And we want to ask, how are these two representations actually related? Well, we can do this, we can evaluate this relationship using an encoding model. And here I will tell you a little bit about how we train this encoding model and how we evaluate it. So this encoding model learns a function f that takes as input the internal representations of a model and outputs and learns to actually output the brain activity in a particular XYZ coordinates of the brain or a particular time point in the brain as well. And so this function f, we need to parameterize this function to directly learn it from the brain data. So we often parameterize this as a linear function, and this is done very often, even in this, going back to this classical work that I presented from, for example, Tom Mitchell and Kendrick Kay. And this is an assumption that has stuck around for many years since then. But some people have tried to understand what are the considerations for having this linear f versus nonlinear f. And actually, they show that there's many different desiderata that we have when using an encoding model. So for example, we want the encoding model to predict very accurately. We want it to be interpretable. And we also want some biological plausibility at the end of the day. And here they show that really, contrary to what somebody might expect, these different desiderata don't actually map onto the linear nonlinear divide very clearly. And instead, this like different desiderata can really refer to multiple research goals. And so each one of those imposes their own constraints on this encoding model. And so they really argue that one should choose a linear or nonlinear model that's really specific to the research question that the person is investigating. And so what we have seen in our work, and a lot of other works have shown this as well, is that a linear model is sufficient to show a significant correspondence between these representations from the models and the brain. And it is at the end the simplest model we can use. So a lot of the work that I'll present today actually makes this linear assumption. But I did want to mention that there are people who are thinking about nonlinear functions f. So here, how do we actually train this function? Well, we want to learn the parameters of this linear function directly from the data. And we use simple machine learning techniques here. So we use cross-validation, and we regularize the learning, usually in order to avoid overfitting, because in a lot of data sets for brain science, there's not a huge amount of samples. So we want to make sure to regularize the learning. And usually people use ridge regression here, but of course you can use other kinds of regularization like lasso or elastic net, but ridge is really the most common one. So once we have actually learned the weights of this encoding model, we want to evaluate how well are we actually predicting the brain response. And we do this in 3 steps. So the first one is to make predictions for held out data that we have never seen during training. And of course, this is very important in general for machine learning. And it's sometimes is overlooked in the brain sciences, but we want to make sure that we make this clear here that encoding models that are done in the usual way do in fact make predictions for this held out data. So this is the first step. And the second step is once we have predictions that correspond to this held out data for each XYZ coordinate in the brain, we want to see how well did we actually predict this data. So what we can do is we want to compare these predictions for the XYZ coordinates with actual true brain response that corresponds to this the sample that we're using to predict the brain response. So in fact, what we can do here, we can use simple correlation metric, so Pearson correlation, that looks at what is the correlation between the predicted and the true values for the different test samples in the data. So once we have calculated this Pearson correlation, let's say we have a correlation of 0.3 or 0.4 for a particular XYZ coordinate in the brain. What does that mean? Does that mean that there's a significant correlation between the brain response and the model? Well, in order to actually answer this, we need to do stringent statistical testing to actually say whether there's significant correlation between the 2 responses. And usually this is done via a permutation test, which I'll be talking to you a little bit in a second. And there's a correction for multiple comparisons because we do these different tests for multiple points in the brain that are correlated with each other. So now I just wanted to go a little bit deeper into this training setup here. So again, we want to find a mapping from the stimulus representation to the brain data that actually generalizes to new brain data out there. So we want to make sure that we can even do this in a zero-shot setting where we can make a prediction for some brain response that corresponds to some words or some images or some stimuli that we have never seen before. And so here the method is we need to split the data set into a train, validation, and test set, which of course, I'm sure many people in the audience are already familiar with this from the machine learning background. And we definitely want to employ a cross-validation to select the model parameters that are based on this validation data set. And again, we use regularization to avoid overfitting here. And the most common technique that we use is rich regularization. And one thing that I wanted to make clear here is that many of the works that I'm talking about today are actually training independent models for each participant that is in the brain data set. So in order to actually make a statement about the brain of general population, we need to test multiple participants. But the way that these models are trained usually is independently for each of these participants, and then the actual results are aggregated across the different participants in the data set. So for example, if we had n participants in a particular data set, then we would train an encoding model for participant 1 and a different function f for participant 2, and so on until participant n. And another thing I wanted to make clear here as well is that very often these works also train an independent model for each different XYZ coordinate or sensor time point in the brain. So there will be a different function f for, let's say, voxel volume pixel 1 in participant 1, and different function f for voxel 2 in participant 2 and participant 1, and so on. So we have these many function f's essentially that are all trained in an independent way. And so this, you can see it as both a limitation and a benefit. So the benefit is that whatever findings, if we have done significance testing and we have corrected for multiple comparisons, whatever findings are significant at the end of the day are in fact found in many different participants independently, in many different areas independently. So this can serve to make us more confident in these findings. The downside is that because there's some signal that's common of course, there's signal that's common across these participants and across different volume pixels and different XYZ coordinates that are nearby each other. And we can leverage these kinds of commonalities to actually get a better prediction of the brain response if we use these commonalities in the signals. And there's some follow-up work here that does in fact take advantage of this. And I just wanted to make you aware of the very standard choice here that a lot of the works that I'm going to tell you about are actually making. So now let's take a little bit a deeper look into the actual specifics of the encoding model, especially when we use it for one of the most prevalent imaging techniques, which is fMRI. So there's a few different oddities for fMRI that are very important to take into account when building an encoding model to predict the activity that's elicited and recorded by fMRI. So the first oddity here is that we very often are actually presenting the stimulus at a rate that's faster than the recording from the fMRI. So let's say if you're listening to a story and I'm recording your brain activity in an fMRI, then you're going to be hearing the story, or even listening to me speak right now. You're hearing a lot of words within, let's say, a second or 2 seconds. but the actual fMRI image is taken every second or 2 seconds, depending on the strength of the fMRI machine. So some of the faster methods can actually take images every second. And in that second, you hear, let's say, 4 or 5 words that were already said. And so the actual response that is measured by fMRI is some kind of function of all of these words that you have heard. So there are ways to actually account for this. So the first one is we want to downsample the actual representation from the model to match the sample rate of the fMRI. And this can be done in several different ways, and it depends on the rate of the presentation of the stimulus. So for example, if the stimulus was presented for the same amount of time, so each word, if, for example, you're reading the words instead of listening to the words, I can present each word to you for the same amount of time, let's say 500 milliseconds. So then your brain response will be more evenly distributed across these different words. And we can downsample the representation for these words by simply averaging all the different words within a specific time window, the representations for these words. But very often we actually use more naturalistic setups in which, let's say, you're listening to the stimulus, in which case each word has a different duration. And so your brain response actually relates to these different durations that you listen to these different words. So then we need to actually take that into account, and we cannot simply average the different representations for these words. So the second oddity that we need to make sure to account for when predicting brain activity that's recorded with fMRI is the fact that the actual brain response that's recorded by the fMRI is a little delayed and it's smooth. So here what the fMRI actually records is what's called a blood oxygenated level dependent signal, or BOLD signal, which relates to the amount of blood that floods into a region and comes out of a region when the region becomes active and then is not active anymore. So here the point is that when a particular region is involved in the processing of some information, it needs more blood because there's a metabolic demand there. And so blood floods into this region, but it takes time for this to actually happen. And so there's a little bit of a delay there. It's about 6 seconds between the onset of the stimulus and the peak of blood the peak of blood volume that's rushing into this region. And then there's also a little bit of a delay until the blood comes out of this region once this region is becoming less active, and that's another 6 seconds. So it's about 10 to 12 seconds from baseline to baseline after a region becomes active. So we also have ways to account for this in encoding models, and this is done in several different ways. So for example, one very popular way is to convolve the representation of the stimulus for particular fMRI image with an actual canonical response function, which is called the hemodynamic response function. And this can give you a more smooth and a delayed signal, essentially, that we can use then as input to an encoding model. But that actually assumes this canonical shape for all of the different volume pixels in the brain. But there's actually work that shows that different regions in the brain might exhibit different hemodynamic response functions. So instead of assuming one particular one and convolving it with the representations from that correspond to the stimulus for that image, another common approach is to let the encoding model learn this function on its own and provide as input the features that correspond to the different images, the different time points in the stimulus that we expect to be related to the actual brain response. So for example, as I said, it's about 10 to 12 seconds of stimulus that may actually correspond to the brain activity for that stimulus. So what we can do is we can provide the features to the model as input that correspond to these last 10 to 12 seconds of the stimulus, and then let the encoding model on its own learn the different weights that correspond to how best to predict the brain response. So this is a very data-driven approach, and it's quite common in the more computational methods that build these encoding models. So now to move on to the actual evaluation setup for this encoding model. Well, as we said, we want to, of course, evaluate on unseen brain recordings. And we do this by applying the learned function, f, to some held out features, held out stimulus features. And we actually get the predictions for this held out brain data. And we want to compare these predictions of the brain data to the true brain data. And there are several different evaluation metrics for this. I talked about the most common one so far, which is Pearson correlation, and I'm showing this on the left side here of the slide, which again uses very simple correlation to look at how similar is the prediction for a particular XYZ coordinate in the brain with the true actual brain activity for the same XYZ coordinate across many different test samples. And this can give you an understanding of how correlated are these predictions and the true brain data across the whole brain for many different test samples. So this is one very popular way of doing this. Another one is to actually have turn this into a classification task. So one such metric is what's called 2v2 accuracy. And the idea here is to actually say which of two possible brain predictions relate closer to their truths. So for example, if you have two pairs of brain prediction and the corresponding truth, we want to see is the actual prediction closer to its corresponding truth or the other truth for the other prediction. And here the accuracy is 0 if both of these predictions are closer to the foil true, and are 1 if both truths are closer to their actual corresponding real brain activity. So this is a way to actually turn this into a classification problem, and this has been used by several studies as well, and it can be beneficial if you're doing these encoding models for data that's quite noisy, so what's called single trial data, in which there's no repetitions of any particular stimulus, and you want to do 0 shots predictions for some new stimuli as well. But both of these, I would say, are commonly used, and the Pearson correlation in recent years has gained more traction than this classification approach. And then the last part is we want to actually do a statistical significance test to understand, are these evaluation metric scores that we got at the end, are they significant? Do they mean that there's a significant relationship between the brain and some model representation? And so here there's a very simple method to do the statistical test, which makes no assumptions about the underlying distribution, and that's a permutation test. And perhaps many of you are already familiar with this, especially if you're coming from a statistical background. But for the rest of us, I will go through the setup for permutation test because it's quite useful, because again, it's non-parametric, doesn't make any assumption about the underlying distribution, and it can be done in a very simple way for many kinds of data. So here, what we want to do is we want to break this input to output correspondence by permuting the output labels. So there's some X, which is the brain activity, and some Y, which is the output labels. the X is the stimulus representation, which goes into the encoding model, and the Y is the brain response that corresponds to the same samples. And so we want to break this input to output correspondence. So we want to permute the Ys, which are the brain activity measurements for each sample. And we're going to estimate the similarity between this permuted labels and the non-permuted inputs, which are the stimulus representations. And we do this thousands of times, which helps us estimate the null distribution of the similarity. So here the thought is that once we permute these labels, whatever similarity we get from our evaluation metric at the end of this permutation is due to chance, right? So there's no true relationship between the inputs and the outputs anymore because we've permuted the labels. So this actually allows us to estimate a null distribution for what the similarity would be that's due to only chance. And then we can estimate a p-value based on this null distribution, which corresponds to the proportion of times that the similarity metric from the permuted labels. So this, again, we did this thousands of times. So it's how many times of those thousands of times did we get better similarity metric, higher score of similarity than this original similarity score from the original unpermuted labels. So this is in fact corresponds directly to a p-value. And this is, again, is very useful tool, and I very much recommend using this kind of permutation test in also different settings that are beyond brain activity prediction. And then specifically for fMRI, something that we need to be aware of is that we need to permute these labels in blocks instead of each sample separately, because fMRI, as we said, has this very slow response, which takes time for blood to go into a region and come out of a region. And so there's some correlations between consecutive images in the fMRI that if we break them using this permutation, if we permute all the samples, all the images, we break this autoregressive structure, and this will unfairly result in a very low similarity score at the end. So we want to preserve this autoregressive structure, so we permute these labels in blocks rather than this independent samples. And of course, at the end, we want to correct for multiple comparisons because, again, we do these independent tests at each different XYZ coordinates in the brain. And these, especially nearby XYZ coordinates, can be quite correlated because let's say they're part of the same brain region, and this brain region has a few specific functions, and it processes some specific information. And so the actual activity in nearby XYZ coordinates can be correlated. And so if we do independent tests for correlated data, we are very likely to get a false positive just because we did many different tests for correlated data. And so here we want to correct for this multiple comparisons of correlated data, and we can do this with standard statistical techniques like FDR correction, so false discovery rate correction, or the family-wise error correction, and so on. And I also urge you to look into this, especially if you're working with very correlated data and you're doing a lot of independent tests. So now we have trained the encoding model. We have evaluated the actual similarity using this encoding model between the model and the brain. And we have gotten some similarity metric score, which we have made sure is significant at the end of the day. And so how do we even look at these scores? So again, it's a very high dimensional vector for all the XYZ coordinates in the brain. So ideally, we would be able to plot it directly on the brain so we can get some kind of understanding that relates to previous work in neuroscience. So there's several different ways to actually plot these scores on the brain. So I'm showing several examples here for different brain imaging modalities. So on the left side, we have fMRI, which I'm showing 3 different ways of plotting both brain activity, but also the similarity scores that we have gotten from an encoding model. I would say my favorite way to visualize is PyCortex, which is this bottom one here. Let me see if I can point on it, point to it. Okay, it's not working for me right now, but it's the 3rd one that's on the bottom right of the fMRI part of the screen. And the good thing about it is it's really improving the fMRI visualization in a number of ways. So first of all, it integrates a number of tools to generate these high quality cortical surface reconstructions. And it uses these surfaces to sample the functional data using a novel projection algorithm that actually results in a much higher resolution of visualizations than other methods to visualize the fMRI. It's publicly available. I've put a pointer to the paper that put it out. And so this is from James Gao and colleagues at Berkeley. And so I highly recommend this tool for visualizing fMRI data. And for if you have other kinds of data, from example, from MEG or EEG, here on the right side, I would really recommend using Mini Python to visualize this kinds of data that has higher temporal resolution, which can, using that Mini Python package, you can visualize both the activity, but also the similarity metrics from encoding models at particular sensors in the brain and also time points in the brain. So again, I think this is quite a nice package, and it's openly available as well. So these PyCortex and Mini Python would be my recommendations if you're looking into how to visualize this performance of an encoding model. Okay, so then this was the first part, which talked about these classical findings and the actual pipeline for evaluating the similarity of representations from models and from the brain. And I want to pause here briefly to see if there's any questions about these very classical findings or the common approaches before I get into the actual more recent findings that use deep learning. So please speak up. So Abhirama has a microphone that he can give you. And Abhirama, maybe you can tell me whether there are any questions or not.
Yes, sure. Any questions? Okay, Mariya, seems like no questions. I think we can proceed.
Okay. Sounds good. Yeah.
Yeah, I think we should have like quizzes after, presentations so that people are motivated to ask questions. Folks, we understand you have just had lunch, but yeah, let's participate. Yeah. Right. Mm-hmm. So if you have questions, please feel free to ask. OK, please interrupt in between as well if you have questions.
Yes, for sure. Please, especially right. So I think these things may be new to many of you, and there might be some questions also at the end that you develop as we go through these more recent findings. So please feel free to ask about any part of the presentation at any point. Just interrupt me. Okay, but for the time being, I will keep going here. So now we'll talk about these more recent findings that actually use deep learning approaches for encoding. And here we use stimulus representations of stimuli from deep learning systems. And I'll focus again on these 3 domains. So in language, I'll be talking about a number of works that date back actually quite early from 2014, which used a very much simpler language models, which were based on LSTMs back in the day, and up to more recent work, which uses transformer models to get the stimulus representations of these naturalistic language stimuli that would present to people in a brain recording device. And the second one is, the second domain is vision, in which I'll be talking about, again, some very, not so recent findings at this point from 2014, which used more CNN, like convolutional neural network-based representations, up to more recent advances that use representations from self-supervised models that are based on transformers. And then at the end, I'll also talk about work in the audio domain, which is a bit more recent and also makes progress using different kinds of speech and also language models for audio stimuli. So first, to focus on this language domain, which also I have to say that the language is my domain as well. So I work in language processing and understanding how representations of language relate in the brain to representations in language models in machines. So this is kind of my area of expertise. So I will talk a lot about areas here in language, but I did want to mention that there's works in vision and audio as well that are very interesting and important, especially if there's people in the audience who are interested in those areas. So here, the first work that I wanted to mention really dates back to quite early. So this is 2014. This was a work at EMNLP. And in this work, the authors here, this is from Leila Webbe and colleagues, and the authors presented a chapter of Harry Potter to a person in an MEG device. So they scanned their brain activity as they read a chapter of Harry Potter word by word. And the stimulus representations that they used were derived from a recurrent neural network, which was actually trained on Harry Potter fan fiction. So the model had not seen the actual book from Harry Potter, but it was trained on a lot of text from Harry Potter fan fiction. And this is, it's from 2014, so this predates pre-trained models. So the authors here had to work hard to create a model that had some understanding of the stimulus of Harry Potter that was more general and had good understanding of the statistics of Harry Potter without using the actual Harry Potter book. And so here, what they focused on is a few different points in time of processing. So again, they're using MEG, which is a brain recording that has a very high temporal resolution. So as we mentioned in the early part, fMRI is much more slower and has a delay that occurs due to this blood rushing in and out. Well, MEG actually relates to the synchronized firing of neurons as this region becomes active. So it's much more quick response than fMRI. So it's actually instantaneous and has a millisecond level resolution. And so here they're focusing on 3 different points in time of somebody processing a word. So in the beginning of processing the word, there are some features that of the brain response that relate to actual inspection of the word form. So this is not about understanding the meaning of the word, but it's more like understanding the word form. And then the second part is the second one, which occurs more towards 200 milliseconds after you read a word, is about judging whether this thing that you're seeing is a word or a non-word. So it's slightly more semantic than the first stage. But then really the last part of processing is around 400 milliseconds after somebody reads a word, which is really about understanding the semantics of this word. So it's understanding what the word means. And so they're going to use different features from different levels in this recurrent neural networks from different layers to predict time points, these different time points in the brain response. So here what they see, they're using these 3 different features extracted from the model. The blue one is the hidden layer. So this is an internal representation from the recurrent neural network. Then the green is the actual output probability from the network, and the purple one is the embedding layer. So this doesn't have any context. So what they show is there's significant word-by-word alignment between the MEG response and the representation of both embeddings. So these are the word level representations that don't contain the actual context, and as well as the actual contextualized representation, which are from the hidden layer of this recurrent neural network. And so they showed this for the first time, that we can use these representations from recurrent neural network, though simple, of course, recurrent neural network by today's standards, to predict image response of people processing words or processing a whole chapter of a book word by word. So this was very exciting. Right. Just a second here. Right. And so here also within the language domain, a few years later in 2018, Shaylee Jane and Alex Huth did another experiment with some stimuli which were actually people listening to a story. But here they used some self-supervised text language models, which were a little bit more recent. So this is 2018, so they were based on more recent LSTMs. And they were trained to predict upcoming words in some other radio stories. And what they showed here is that they can vary the amount of context that they present to the model. So for example, they can show either 1 word or 5 words or 10 words or 15 words. And by increasing the amount of context they present to the model, they're able to predict the brain response better also. So this contextualization of the words actually help predict the brain response better as well. And what they found also is that the middle layers, so they used the 3-layer LSTM in this work, and what they found is that layer 2, so the middle intermediate layer, was the one that predicted the brain response the best. So this is what's shown here in purple or pink on this plot. So we see the highest ability to predict the brain using the middle layer of the model. And this was very interesting because more earlier work, especially in vision, showed that when using supervised models that are trained for, let's say, object recognition or scene classification, the late layers in these models actually corresponded best to some high-level brain regions that are involved in semantics. So here, what these authors showed is that actually the middle layers of self-supervised models are the ones that align best with the brain response during high-level semantic understanding. And this is thought to be due to the fact that these models are trained in a self-supervised way. So both the input and the output of these models have information that's related to the word level. And so the middle layers may be the ones that contain information that's the most high level and more similar to the brain response. And we actually, in some of our work, we've looked a little bit into this this thought, and I'll be presenting this work in the later session, the next session, which is on advanced methods in brain encoding. And so stick around if you want to find out more about this, the reasons behind this alignment in the middle layer between the brain response and the models. So again, what this work showed is that there's significant alignment between a bit more recent NLP models and the brain, especially when we vary the amount of context, and this best alignment occurs with the middle layers of the models. And so, in much more recent work that actually used the pre-trained NLP models, which is actually work from us. Here we looked at some pre-trained natural language processing systems, which were pre-trained on large corpora. So this included BERT. So this is in 2019. So BERT had already come out. I'm sure you're all familiar with this model. And here we focused on specific regions in the brain that are thought to process more long-range context versus short-range context. And we looked at how different types of NLP systems, pre-trained NLP systems, were actually able to explain these short-range or long-range regions. And we looked at 4 different kinds. So here we looked at ELMO, which is LSTM-based, and a few other ones like BERT and Transformer-XL. And Transformer-XL was actually specifically proposed to be able to encode larger, longer-term contexts. So what we actually found was that indeed Transformer-XL was able to maintain the most context in terms of as we provided more and more context to Transformer-XL, that was the only model that continued to predict the brain response better and better. Of course, up to some point, only about 50 words or so, which is much shorter than the range, the long range that Transformer-XL should be able to maintain. And we also had findings that aligned with these previous findings about the middle layers of the LSTMs aligning with the brain response the best. We also found that middle layers in transformers aligned best with the brain response. So we found that across several types of large language models, the best alignment with fMRI was in fact in the middle layers. And so one more work in this direction, in the follow-up work from a different group. Here they looked at sentences specifically, and they contrasted the MEG versus fMRI response during reading as well. And here they also used the different kinds of models. And again, they found that the best alignment with both fMRI and MEG is occurring in some of these middle layers. So some of the models that they use here are autoregressive models, so based on GPT-2. And what they also found that was very interesting at the time was that the better a model was at actually predicting the next word, the better its actual prediction of fMRI and MEG was. So they found a correlation between the ability of the model to perform next word prediction and its ability to predict the brain response. And there's a few works in this line as well that have shown that this for a broad range of models. So this is work at PNAS by Martin Schrimpf and colleagues. And here they show that both in fMRI and ECOG, which is another type of brain imaging, and in reading and listening, they find this correlation between the ability to predict the brain response as well as the ability to predict the next word. And additionally, what this work offered was they were able to estimate how much of particular brain activity in some region should be explained. So they were able to estimate the explainable amount of variance in a particular brain region. And what they showed is that some models at the time, so this is work from 2021, so they found that GPT-2, which was one of the newer language models at that point, was able to explain up to 100% of this estimated explainable variance in some specific language regions. So this is not all language regions. So if you actually look at it more broadly, we see that these kinds of models can explain something like 60 to 70% of the explainable variance across all language regions. But in fact, in some particular small language regions, we can explain up to 100% of this estimated explained variance. So this, of course, was very interesting, and it means that we have made a lot of progress with these language models, but still there's about 30 to 40% of variance in the brain response that we are yet to actually measure. And so one last work in this language space is actually showing even more evidence for this ability to predict the next word of a model as a key mechanism for alignment with the brain. And here what they show is that the representations from language models can predict brain activity for upcoming words. So there's some correlation between what words was presented now with what word will be presented, the activity of the brain in the future. So here they're making a statement about the fact that the brain is also possibly doing prediction of what's going to come next, and this prediction is what is aligning the model to the brain. There has also been more follow-up work since then, showing there's other mechanisms of alignment as well, in addition to this prediction. But this prediction does seem to explain a substantial amount of variance in this ability to predict the brain with the language models. So we're running a little bit low on time, so I'll go through this vision part, and perhaps we're not going to have time for the audio. I'm very happy to discuss this more if people would like in the break, but I know that you must also be losing a little bit of your attention after lunch, and so a coffee break will come in handy. But I wanted to go through this vision part as well. And so for this vision part here, I wanted to talk to you about work from 2014 that, so it's quite old at this point, but this was the first work in the vision domain, which used layers in a pre-trained convolutional neural network to predict different responses in a monkey's brain during processing of natural objects. So what they focused on was some specific regions in the monkey's brain, which are known to participate in visual processing. And in vision, we know that there is a hierarchy of processing that goes from early visual processing to late visual processing. So the early visual processing is more related to this like V1 to V4, and the later visual processing is related to the IT region, which is inferior temporal cortex, and this is the highest cortical area in the ventral stream. So very briefly here, what they found that they looked at different layers in a supervised vision model, and what they found is that the highest layers in these supervised vision models were most predictive of this highest layer or highest level area in the vision ventral stream, which is IT. And the intermediate layers in the model were most predictive of V4, which is a more intermediate stage of processing in the visual cortex. And then there's some additional work after that, which also looked at fMRI and MEG together and tried to explain actual human brain response to these different natural objects. And here again, they see very similar results, which is that a CNN, a convolutional neural network that's tuned for object classification, is capturing these stages of human visual processing in both space and time. So this is work that adds to this understanding of the information processing across a hierarchy of areas. And now some more recent work which uses self-supervised models. Here what they showed is that they used representations from contrastive learning, a contrastive learning approach to explain fMRI responses of people processing different images of objects. And what they found briefly is that using these self-supervised representations can achieve similar ability to predict the brain response to get to as the category specific models, as the task optimized models along this visual hierarchy. So this was interesting as well because it showed that even self-supervised models, the supervision with specific objects, object classes is not necessary to actually achieve the ability to predict the brain. And in this last work that I wanted to mention in this vision part is that also some work that occurred at the same time looked at how well we can align these representations from self-supervised models again with different recordings from monkeys in the visual domain. And here what they also show is that the self-supervised models can produce brain-like representations. And the really cool part here that added to previous work is that they train these self-supervised models using data only from data that was from head-mounted cameras on children. So this is data. This is as though the model saw the world as a child through the development stage. And the model was only trained using this very noisy data from head-mounted cameras from children. And even so, still this representations that it was able to build internally related very much to the actual representations in a monkey's brain as it was seeing different images. So now, again, for the, because we're running low on time, I wanted to go back to this agenda and leave a few minutes for questions and allow you to do your coffee break. And so here, again, we talked about deep learning for brain encoding in the session. We talked about some classic findings and some of these common approaches for how to actually build encoding models. And we discussed some recent findings that use deep learning to extract representations from the models and actually predict the brain response to very naturalistic stimuli. And so this was the presentation for the brain encoding part. I'll be back in 30 minutes to discuss with you some advanced methods in brain encoding and decoding. And I look forward to having you back here as well. But for now, I would love to answer any questions that you might have on the brain encoding part, any part of the presentation.
Yes. There's a question from one of the audience in this game.
Yeah. Hello, Mariya. Can you hear me?
Yes.
Okay. Can you please briefly touch on the audio part of the current new development on the audio parts? You choose to skip.
Okay, sure.
So I'm not eager to have a coffee yet.
Okay, sounds good. How about this? How about we take questions now, and then in the advanced methods in 30 minutes, in the beginning, I can go over the audio part. Would that work for you?
The problem is that I have to leave after the coffee break because of another meeting.
Okay. Okay, sounds good. So I will briefly touch on that. Let's see. So here for the audio part, here in this specific model, they used fMRI responses to people seeing natural sounds, and they use deep models optimized for both speech and music recognition. And here what they found is, so again, they looked at different layers of this network that's optimized for speech and music. And they looked at how the brain response is predicted well by different layers in the model. And what they showed was that there's some primary auditory responses that are predicted best by this intermediate layers of task-optimized models. And the non-primary responses are predicted best by late layers. So this again matches this understanding that in a task-optimized network, the higher layers are match more with higher levels of higher areas of processing in the brain, and the earlier layers and the intermediate layers are more like the early processing in the brain as well. And just maybe one more work here is from a more recent work that uses self-supervised speech models. So this is like Wav2Vec, for example, if you're familiar. And they looked at how can we predict the brain response and the function of the representations, again, from different layers of Wav2Vec. And what they showed was that, again, the middle layers of the self-supervised models predict best the auditory cortex in the brain. So here, again, it's more like the language models, which are self-supervised, in which the middle layers are the ones that really predict the brain response the best, whereas the early layers are the ones that predict more the early auditory cortex better. And so I would say that's a good summary of the work in this auditory domain.
Right. Just to add, Mariya, right, I'll also add in parallel interspeech is happening. And at interspeech, we have a paper where we also have done some of this audio work. I think just where Mariya left, we have also taken models which are fine-tuned on the Wav2Vec model, Wav2Vec 2.0 model, fine-tuned on several kinds of speech tasks, like automatic speech recognition, keyword spotting, speech emotion recognition, speaker identification, and so on. And we have tried to see which of them are best aligned with the brain activations. So exploring, sort of audio encoding from fine-tuned Wav2Vec 2.0 kind of models perspective as well. So hope that helps you with your question.
Thank you.
Thank you. Yes.
Any other question? Yeah.
Am I audible?
Yes.
Yeah. So I also wanted to ask regarding the recently released Things dataset, like uses multimodality. So how can we use it to get better brain encodings, like object representations?
I see. So things. So in general, the way we can use multimodal representations in the brain is to also use multimodal models. So for example, something like Merlot Reserve, if you have heard of this model, or Video BERT as well. There's models that take as input actual video frames and auditory input, and even the transcript, the speech that's happened in the video is input, and then combine all these representations in a joint representation. You use this representation as input to an encoding model. So for things specifically, so the things dataset that I'm familiar with presents all images of objects. So I would not call that multimodal. I would call that images, right? So for that, I would use a vision-based representation rather than a multimodal representation. But there's other data sets like, for example, Friends. So some recordings from Friends, the TV show. So people watching Friends or people watching movies. And so those are truly multimodal encoding in the brain. And so I would use a model that has a multimodal sensory representation through the encoding model.
Okay.